Hey guys, welcome back. Today we are going to cover chapter 12 from Campbell's Biology in Focus. It's going to talk about the chromosomal basis of inheritance. So we'll start off with an overview. So locating genes along chromosomes. So Mendel's hereditary factors, what he called genes, we now know are genes, and we know that genes are located on these things called chromosomes. Okay, so the location of a particular gene can be seen by tagging isolated chromosomes with a fluorescent dye that highlights the gene. So essentially, you can tag, I'm going to draw, you know that I'm not very good at drawing, but you can tag different genes to glow different colors on chromosomes to view under fluorescence. So that's one way that we can track, is this gene on this chromosome? If yes, where is it? Okay, so starting off at the top, so Mendelian inheritance has a physical basis in the behavior of chromosomes. So we learned about mitosis and meiosis. They were first described in the late 1800s. So the chromosome theory of inheritance states Mendelian genes have specific loci or positions. A locus would be one, loci, many positions, on chromosomes. Chromosomes undergo segregation and independent assortment. So these we talked about previously, the laws of segregation and independent assortment. The behavior of chromosomes during meiosis can account for Mendel's laws of segregation and independent assortment. Okay, so here's kind of an overall picture of all of this. So we've previously talked about the different types of generations. We see over here the law of segregation, and over here we see the law of independent assortment. So just as a refresh, the law of segregation has two alleles for genes, has two alleles for each gene that separate. So if you have a homozygous dominant, then that's the only gene that you can actually get from your parent. If your parent is cool and they are heterozygous, then you have a 50-50 chance of inheriting either one of those alleles. For the law of independent assortment, the alleles on the genes are non-homologous. On non-homologous chromosomes are going to assort independently. So this just means that it doesn't matter if your height and your eye color are inherited together. They're on different chromosomes. They're non-homologous, so they're not going to necessarily go in the same gamete. Like, just because you have blue eyes does not mean that you also have to have freckles, right? These two things are not dependent on each other. So that's why they independently assort. So they can go into the different gametes at random. So we're going to talk about this guy, Thomas Hunt Morgan. So Morgan's experimental evidence through scientific inquiry. So he also had a huge role in what we know today of genetics. So Thomas Hunt Morgan and his students began studying the genetics of a fruit fly, and they're called Drosophila melanogaster, and they're disgusting. And when you get to genetics in college, you're going to have to deal with them a whole bunch, and you have to grow them, and they smell, and it's great. Okay, but they studied fruit flies in 1907. So there's several characteristics that make fruit flies a convenient organism for genetic studies. Previously, when we talked about Mendel, we talked about his pea plants. So they were, fast growing. They weren't living beings like animals, right? They're plants, so it's a little bit more just to be replicating plants, right? So now we're moving up to fruit flies here. Okay, so they're convenient just like plants because they can produce a lot of offspring, and a generation can be bred every 2 weeks, which is very fast compared to, think about a human, where it's a little bit unethical, but you're also gonna have to wait a year almost, right? And then they only have 4 pairs of chromosomes. So the chromosome number is very low, so it's easy to track things. Okay, so Morgan noted a wild type, or the normal phenotype, the normal characteristics of these flies that were common in the fly populations. So the traits alternative to the wild type were called the mutant phenotype. So basically you have your normal and then your mutants. He had these 2 classes. Okay, so the first mutant phenotype was discovered in a fly that had white eyes instead of the wild type, red. Okay, so we first discovered this first mutant that was white eyes instead of the wild type, which is red. And we'll look at a picture. So their eyes are actually physically different colors. So that's what we're talking about. So again, our wild type is normal and wild type is red. So this is normal. And then this little guy has a mutation. So that's our 2 groups, normal, mutant. That's the first mutation that was discovered. Okay, so we're going to look at correlating behavior of a gene's alleles with behavior of a chromosome pair. So in one experiment, Morgan, who's working with these fruit flies, mated male flies with white eyes, the mutant type, with females with red eyes, the wild type. The F1 generation, their offspring, right? So we have this male and this female, and when they have a batch of baby flies, this is the F1 generation. They all had red eyes. So that tells you that there were no white eyes, right? So they all have red eyes. The F2 generation, when we take all of these that have red eyes and mate them together, get the F2 generation, showed the classical 3-to-1 red and white phenotypic ratio there. That's the word, phenotypic ratio. But only males had white eyes. Interesting. Only the males had white eyes. So Morgan concluded that the eye color was related to the sex of the fly because he only saw the mutation occur in the males. So Morgan determined that the white-eyed mutant allele must be located on the X chromosome. This is something that we see commonly with sex-linked traits, that they are all on the X chromosome. So Morgan's findings supported the chromosome theory of inheritance. So we're going to continue to talk about all of this and what this means, but this is something important for you to remember, that if there is some sort of sex-linked characteristic that males are more likely to have a disorder or some sort of trait than a female, it's because it's located on the X chromosome. Okay, so here's the experiment overall that we just talked about. So the experiment, the parental generation, you have your females having the wild type and your males with the mutant. All of their offspring had the red eyes. And then when you bred one of these with another one of these, then you ended up with the F2 generation, which gave you the classic 3-to-1 phenotypic ratio. But notice here that we have a male that has white eyes, which is an indication that this is perhaps a sex-linked trait. Okay, so the other side here is showing you the conclusion that the P generation, we have our, you can see the chromosomes here, the X and the Y with the traits that they're carrying. You can see this on the chart here. Hello, rude. You can see this on the chart here with the sperm and the egg. You can see that we have our females and our males. And then, so these both have the, one characteristic from each parent. And then in our F2 generation, when you have two of these F1 generation individuals, hello, rude, together, that you can see that we have our our one mutant type male here. So that is the overall conclusion of his findings. And I said that this is more common when we have males are more common when the males are more likely to have this trait. It's because it's a X-related chromosome abnormality or trait. Because remember that females have XX and males have XY, right? So if you have one X that carries the trait in a female, the other X is normal. So typically that just means that this female would be like a carrier for a trait. She wouldn't actually have it. Both of her X's have to have that trait in order for her to show that trait. So if she's just a carrier, she doesn't show it. But if she has both X's affected, that means that she's going to have that trait. Whereas you look at the males on the other side of things, if they only have one X and that one X is going to show that trait, then that means that there is no other X to cancel it out as a backup. That's all that they have. So if they have an X that has the trait on it, then they're going to show the trait. Whereas if they have a normal X that does not have the trait on it, then they'll be normal. Just like this guy here and this guy here. This one is normal. This one is a mutant. Okay. So let's talk about sex-linked genes and how they exhibit unique patterns of inheritance. So in humans and some other animals, there's a chromosomal basis of sex determination. So this means that we have females and we have males, and that's XX for females and XY for males. Biologically speaking, that is how sex is determined. Okay, so the chromosomal basis of sex. In humans and other mammals, there are two varieties of our sex chromosomes, the larger X chromosome, and I mean larger. It is much larger than our little Y chromosome. It's a lot larger because it carries a whole lot more information, and a smaller Y chromosome. Only the ends of the Y chromosome have regions that are homologous with corresponding regions of the X chromosome. So they have little in common, meaning that they don't carry much of the same information at all. And the SRY gene on the Y chromosome is required for the development of the testes. So the Y chromosome is literally what makes a male, biologically speaking. Okay, and when I was learned this, wow, that is not grammatically correct. Words are difficult today. It's been a long day. The SRY gene, when I was taught this as a freshman in high school, my teacher was like, you know how, males are always like apologetic to their wives because they're in the doghouse a lot sometimes. Males have the Y chromosome because they're always really sorry. And that's a really bad joke, but maybe that'll help you remember it. Okay, so the Y chromosome carries the SRY gene that allows the development of the testes, which is, of course, the male reproductive organ, because that's where the sperm are created, right? So that's what makes a man. This is the difference. Like I said, the X is a lot larger. Think about if all individuals have an X chromosome, it must have something important on it. And it's going to be some sort of sex-determining characteristics, but also there's going to be other genes encoded on such a large chromosome, right? Whereas the Y, literally the most important gene there is going to be the SRY, which is going to make an individual a male versus a female. So you can see that it's a lot smaller here. So we know that females are XX and we know that males are XY. We've been talking about this for a while. So each ovum contains an X chromosome. This means that each egg that a woman produces has to carry an X chromosome because that's all she has. She has an X and an X. She can't donate anything else because she doesn't have anything else. Okay, and then the sperm is either going to contain an X or a Y because a male has both of these chromosomes. So the sperm is actually what determines the sex of the offspring, because you know that there's already going to be an X waiting there in the egg to be fertilized. Then when that sperm comes in and it carries an X, it's going to be a female. Or if the sperm comes in and it's going to carry a Y, then that's how you make a son or a male. Okay, and then we have other animals that have different methods of sex determination, which just means that maybe it's multiple, not just like one specific sex chromosome. Maybe there's multiple chromosomes that are going to be interacting in order to determine the sex. But we're talking about humans and human genetics. So we're going to be just using the X and the Y. So here's an image of that for you. You know that you have 46 chromosomes in all of your cells. So in your basic body cells, you know that 44 of those are going to be autosomes, which means that they are going to be coding for things that don't relate to your sex, where the last two, 45 and 46, are going to be talking about your specific sex chromosomes. So then, of course, with the females, as I said, she can only donate an X because that's all that she has. And then the males have both the X and the Y. So they're able to donate either of the two. So then you have the option of having a female offspring or a male offspring. Okay, so a gene that is located on either sex chromosome is called a sex-linked gene. That makes sense because it's located on a sex chromosome. Makes a lot of sense there. It's sex-linked gene because it is a gene that exists on a sex chromosome. So genes on the Y chromosome are called Y-linked genes, and there's very few of them. We're actually not even going to talk about them because there's so few of them. The ones that we see a lot more prominently are going to be X-linked genes. So these are genes that are located on the X chromosome, and they are called X-linked genes. So the inheritance of the X-linked genes. So the X chromosomes have genes for many characters unrelated to sex. Like I said, it's such a large chromosome. It's got to have a lot of other things encoded on it rather than just things that determine sex. Okay, whereas the Y chromosome mainly encodes genes related to sex determination. And remember, that's where we have the SRY gene that's going to code for making testes, which will make sperm, which makes a male. So the X-linked genes follow specific patterns of inheritance. So for a recessive X-linked trait to be expressed, the female needs to have 2 copies of that allele. That would be a homozygous female. So she has an X from her mom and an X from her dad that both carry a recessive trait. Therefore, she will have that trait. Okay, and a male, they call this like hemizygous because you only need 1 copy of it because there's only 1 X in the male's sex chromosome makeup. So a male only needs 1 copy of an allele, and that's called hemizygous. So I'll draw this out for you again. But our X-linked recessive disorders are much more common in males than in females. Okay, so there's lots of different ways to denote, like, for instance, color blindness. Sometimes you'll see it as like an X to the C, as having like that's an X chromosome that has the color blindness, and then like this would be a normal X. So sometimes that's how it's denoted, or sometimes because this is here saying that it is a recessive X-linked characteristic. So for instance, you'd have like a capital C like for the dominant, and then you'd have a lowercase c for the recessive. So this female here would be a carrier because she has one affected copy of a recessive X-linked trait. So this says here she needs to be homozygous if she's going for it to be expressed, if she has it. So she would be X to the little c, X to the little c. She would be colorblind. Whereas a male, if he has X to the little c, Y, he only has one X, therefore he's colorblind. Whereas he has X to the big C, Y, right? This tells you that this is not going to be colorblind because it is a dominant trait. So there's different ways that you see it. Like I said, I like this convention the best because I think that it's the simplest, but a lot of test questions, a lot of textbooks use examples where they actually show the dominant and recessive traits. Okay, so this one is showing you the uppercase and lowercase, so dominant and recessive. So you can see here that our female has X to the N, X to the N, so they're both capital, and our male, which is colored in, which means he's the one that is actually affected, X to the little n, so this is going to be a recessive disorder that is an X-linked trait. So our male is affected, our female is not affected. So then when they have offspring, you can see here that these are females, and these are our males. Okay, and you can see that both of the males have a capital, so they are not affected. They're not affected. Whereas these females, they only have one copy. And remember up here, we said that they had to be homozygous, and these are not homozygous. This is heterozygous. So these are carriers. This means that they can pass on the trait, but they themselves do not have the trait. So whatever this is examining here, they are not affected, but their offspring could potentially be affected because if it is a recessive trait, they're going to give one of their kids a recessive trait someday, right? So let's look at that. So if we take these two and these two, we'll look at the left first. Okay, so this is a carrier female. So remember, she's not affected, but her offspring might be. And then you have a normal male that she's mating with. Okay, out of the children, you see 4 different possibilities. You see a normal female, you see a normal male, you see a carrier female who's not affected but can pass it on, and you see an affected male. That's what the coloring means here. White is normal, the light color is a carrier, which is only females because they have 2 X's, and then for our dark colored, that is the affected individual. So also take note here that we have, the options are out of 4, right? But really think about the sex here. There's only 2 females that are possible right here and 2 males that are possible because you have a 50-50 chance of having a son or a daughter because the sperm cell is either going to be carrying an X or a Y. 2 options, that's a 50-50 chance. Okay, so out of our females, 50% of them are carriers. How do I know that? Because there's only 2 of them and 1 of them is a carrier. Out of our males, 50% of the males are going to be affected. Whereas if I asked out of total offspring, 1/4, 25% of the total possible offspring. Does that make sense? So you have to make sure that you understand that there is a female option and a male option, which is 50%, and then you're looking at half of that. Okay, so if it asks you specifically in a question to determine the sex, what percent of this sex is affected, you cannot say out of 4. Okay, that's very important that you remember that. All right, and then our other example on the other side here, you have a carrier female and you have an affected male. You can see a higher prevalence of this trait here. So you have a carrier female, you have a female that is affected because she has 2 recessive traits, and then you have a normal male and an affected male. So again, you'd have 50% of females are carriers, 50% of females are affected. You'd have 50% of males being normal and 50% of males that have the trait, actually. And then out of all of the offspring, their offspring in general have a 50% chance of inheriting that trait. So let's look at some of those examples. You have some disorders that are caused by recessive alleles on the X chromosome in humans. So color blindness is one that we just talked about. And no, that doesn't mean that you can't see color. There's just different variations of color that you have a hard time either differentiating between or you flip them. Then you also have Duchenne muscular dystrophy and hemophilia. So we're going to look at X inactivation in female mammals. So remember that we said that the sex chromosomes for female are X and X. Okay, so remember that in a male, they're X and Y. So in a female, she really has 2 copies of a whole bunch of different genes. It's not just her sex genes. Remember that the X chromosome contains more genetic information than just what's making up your sex, right? So in mammalian females, one of the 2 X chromosomes in each cell is randomly inactivated during embryonic development, is randomly inactivated. Okay, so the inactivated X actually condenses to form a bar body. That's just the name of it. So you have one X that will be expressed, whatever genes are there, and one that is not. And it is random. So each cell can actually have a different, it's kind of like a mosaic, like a different pattern, essentially, of what's being expressed. So if a female is heterozygous for a particular gene located on the X chromosome, she will be a mosaic of that character. So like I said, so if you are a heterozygous individual for a trait, remember that this is heterozygous. So sometimes her cells will show the dominant and sometimes her cells will show the recessive, because it's just depending on which X is activated or inactivated in each of her cells. That's actually how you get the rise of calico cats. So here we go with that example. So you have an early embryo. So you have an X chromosomes. You have the allele for orange fur and the allele for black fur. So in one of these, you have the active X as being the allele for black fur, which means that the orange fur is inactive. And then you have vice versa on another cell. So you have the activated orange and the inactivated black fur. So that's how you get the different colored patches on calico cats. Okay, next we're going to talk about linked genes and how they tend to be inherited together because they're located near each other on the same chromosome. Okay, this is something that gets a little bit confusing, but literally the closer together they are, the more likely they are to be inherited together. That's pretty simple. I'll say it again. The closer together 2 genes are on a chromosome, the more likely they are to be inherited together. Okay, so each chromosome has hundreds or thousands of genes, except for the Y because it's really small. Okay, so each chromosome has hundreds or thousands of different genes. So genes located on the same chromosome tend to be inherited together. They're called linked genes. Okay? If they're inherited together, they're called linked because they go together. It's like a package deal. Okay? So how linkage affects inheritance. So Morgan did experiments with fruit flies to show how linkage affects inheritance of 2 different characters. So Morgan crossed flies that have different traits of body color and wing size. So Morgan found that the body color and wing size are usually inherited together in specific combinations, or the parental phenotype, so whatever the parents looked like. Okay, he reasoned that since these genes did not assort independently, they were on the same chromosome. So again, did not assort independently. This means that they had to be on the same chromosome because remember I said you can have green eyes and be short. You can have green eyes and have freckles. You can have blue eyes and have freckles. Therefore, freckles is not attached to eye color. They're independently assorted. Whereas if he's saying that there is a specific combination, if this body color and this wing size are always together, that means that they have to be on the same chromosome. So that's how we get linked genes. Okay, so here we have the F1 dihybrid female and homozygous recessive male in a test cross, and most of the offspring have the following characteristics. Okay, and these like B and VG, it has to do with the different traits for the wings and the different traits for the body color. You can just see the differences here with the plus signs or the lack of a plus sign. It's not necessary for me that you know what each of these characteristics are because I will not be asking you about this experiment specifically on an exam. Okay, so the experiment. So the parental generation is homozygous. So you have the wild type, which is a gray body and normal wings. And then you have the mutant type, which is a black body and vestigial wings, which means that they're little and they don't really function. Okay, so here's your characteristics that you're looking at with the wild type and the mutant. Okay, with the F1, so their offspring, when you do a test cross with the wild type F1 dihybrids, you can see here that we have these traits that we're studying here. So you have the wild type F1 dihybrid, which is a gray body and normal wings. Okay, and then you can see, that's just rude. Are you done? Thank you. I guess not. Oh, wait. Okay. Then you also have the homozygous recessive, which is the black body with the vestigial wings. So notice that this is exactly the same. And notice that this over here, we're actually missing a plus sign. Okay. But that just means that that's the dominant. So that's okay. And we're missing the B here. So these are actually, these are our heterozygous. This is the wild type F1 heterozygous because it's a mix of the parents. Okay, so when you do a test cross here between these two individuals on this little chart, you have the predicted ratios, the genes on different chromosomes. If that were the truth, then you'd expect 1 to 1 to 1 to 1. But if the genes are on the same chromosome, that now you have the ratio of 1 to 1 to 0 to 0. So then the actual results show us that there's a much higher prevalence of the wild type normal gray body and the vestigial black and vestigial wings with the black body than there are with the vestigial gray, vestigial, and the black normal. So these two greatly outweigh, right? So this means that the genes must be on the same chromosome because these are predicted ratios. And all of this math down here is accomplished by something called the chi-square model. And I'm actually not going to explain that in this lecture. I'm going to do a whole nother lecture about it because it's a little bit complicated. And here I just want to focus on the actual chromosomes themselves. So I will do, my next lecture will actually just be focusing on chi-square. It's really not that complicated. But I deleted a couple of slides in here that followed this that explain that. So I just don't want that to be like a hole in your brain. I'm going to fill that hole in the next lecture. Okay. So basically through math that I'm going to teach you later, we know that the genes if the genes are on the same chromosome, that this is the ratio that they will display. And this is much closer to what we actually saw than if they were on different genes. You can just agree here that this ratio doesn't really fit anything, whereas this ratio looks a lot like what we got in our results. Okay, so however, the non-parental phenotypes are also produced. So understanding this result involves exploring genetic recombination. Okay, so basically we're saying that we got other results that we weren't necessarily expecting, and that's because we have recombinant genes. So this is genetic recombination, the production of offspring with combinations of traits that are different from either of the two parents. So this means that there is an offspring that is produced that doesn't have exactly the same trait layout as the parents. That's okay. It's called genetic recombination. So genetic recombination and linkage. So the genetic findings of Mendel and Morgan relate to the chromosomal basis of recombination, which we're about to talk about. So recombination of unlinked genes is independent assortment of chromosomes. So Mendel observed that combinations of traits in some offspring are different from either parent. True. Okay, so the offspring with a phenotype matching one of the parental phenotypes is called a parental type. Makes sense because it's the same type as the parental. And then offspring with non-parental phenotypes, these are new combinations of traits, are called the recombinant types or recombinants. A 50% frequency of recombination is observed for any two genes on different chromosomes. So it's a pretty high frequency on different chromosomes. Okay, so here's an example of that. So you have the parental types here because this says yellow and round. Well, here's yellow and round. And this one says green and wrinkled, and here's green and wrinkled. But then how did we get these? Right? Well, we know that if we have the genotype here, that the possible gametes are going to be these combinations. The genotype here, this is the possible gamete combination. And when you put them together, these two end up being possibilities, right? Even though they don't look like the parents, it's just how the genes are interacting with each other. It's just the genetic combinations that exist allow us to have these recombinant offspring. So recombination of linked genes and crossing over. So we've talked about crossing over and we talked about meiosis, specifically prophase 1. So Morgan discovered that even when two genes were on the same chromosome, some recombinant phenotypes were still observed. So this is now on the same chromosome. Before we talked about different chromosomes, how there's about a 50% frequency of recombination. Now we're talking about the same chromosome. There's still some. Notice it's not allowed. It didn't say 50% here. It says some recombinant phenotypes are observed. So he proposed that some process must occasionally break the physical connection between genes on the same chromosome. So if they're both, on these chromosomes here, and you've got one that's located here and one that's located here, you'd expect that they'd kind of stay together, right? But because of this thing called crossing over, we know that when you have our homologous chromosomes that are close together, sometimes, their little legs get intertwined and then they swap information. That's crossing over from prophase 1 and meiosis 1. Okay, so that mechanism is called crossing over between the homologous chromosomes. Okay, so this is another image where we're looking at the recombination frequency after this series of test crosses, which we can see here is 17%. Okay, so we're looking at the B and the VG genes. Okay, so those are located on one of these chromosomes, which you can see here. Those are the locations. So we're looking at the recombination frequency is about 17%, which is lower than 50%. So we can start to think it's probably going to be on the same chromosome because, well, here in the picture, you can literally see that it's on the same chromosome. But if you're just looking at the math, that's a lot less than 50%, which means it's probably on the same chromosome, that these two genes exist close together on the same chromosome. Okay, so new combinations of alleles, variation for normal selection. So recombinant chromosomes bring alleles together in new combinations in gametes. So random fertilization increases even further the number of variant combinations that can be produced. So you know that there's random fertilization. It's not like, oh, this one sperm cell has to attach to this one egg cell. Species-wise, yes. But it's not this one sperm cell of the million that were just deposited that have to go and fertilize this one ovum, right? It doesn't work that way. It's completely random. That's why it's called random fertilization. Okay. And then we also have, the recombinant chromosomes that are bringing alleles together in our new combinations in gametes. That's what we're talking about crossing over. And of course, how you have the independent assortment and everything in all of the gametes. Okay. And then this abundance of genetic variation is the raw material upon which natural selection works. We're going to spend a lot of time on this between chapters 19 and 25-ish. Okay, that's going to be all about evolution and natural selection. But this right here, when we're talking about genetics and we're talking about making the next generation, this is the basis on how all of that works. The more genetically diverse a species is, the more recombinant chromosomes you have, the more genetic variation that you have in the species, the stronger and more fit that species is for existence. And we'll talk about the importance of all of that as we move on. Okay, so now we're going to look at mapping the distance between genes using recombinant DNA. So we're going to talk about a new scientist. He's one of Morgan's students, and he constructed a genetic map, which is an ordered list of genetic loci, the locations, along a particular chromosome. So you've got your chromosome, and we're going to be like, oh, gene 1 is here, and gene 4 is here, and oh yeah, we found out gene 2 is over here. You're going to actually be able to map them. Okay, so our new scientist predicted that the farther apart two genes are, the higher probability that crossover will occur between them, and therefore the higher the recombination frequency. Makes sense. Let's think about it. If you have crossing over, okay, I'm just going to draw like a simpler version of this. The crossing over is like I said, when these little legs get tangled and you split and you end up exchanging genetic information. If you have a gene that's right here and a gene that's right here, the odds of some sort of crossing over with another chromosome that's going to split right in the middle right there between those two to separate them, that's not very likely because they're very close together. Okay, whereas let's say that I have another chromosome and I've got a gene here and I have a gene here. Look at all this space, all this room for them to separate because of crossing over. Right? So the closer together they are, there's a very tiny little chance of crossing over between them. Whereas if they're further apart on the chromosome, there's a lot of space there for crossing over to happen, right? So that might actually split off and swap with whatever's here, which means that then those two genes are not on the same chromosome anymore. Okay, so that's increasing our genetic variation, but we can create a genetic map based on the frequency of crossing, or the frequency of crossing over, the frequency of recombination, essentially. Okay, so linkage map is a genetic map of a chromosome based on the recombination frequencies, like I said. So the distances between the genes can be expressed as a map unit. So one map unit represents 1% recombination. So if I have this chromosome and I have a gene here and I have a gene here, and I say that they are 5 units apart, this means that they are 5%, they show a 5% recombination frequency. Okay, whereas like let's say that I have one that has, a 30 units apart, means that these two genes show a 30% recombination frequency. Okay, that means that they're further apart. So the shorter the percentage, the closer together, the higher the percentage, the bigger distance between them, the bigger the distance between them. So here's an example of that. This is actually the results from that fly picture that we just looked at, that I said, I don't care if you know the specifics of it. I just want you to understand what a recombination frequency is and what this map is. And we're actually going to make these in my class with some pool noodles and examine how that looks. Okay. So here you can see that you have the recombination frequencies, and notice, yes, I know that 17 and 18.5 do not add up together. I know that, but sometimes the difference is because of canceling out with recombination. We're not actually going to get into that. I don't need you to know that. Just understand that we're going to say, quote, close enough that we're going to call them the same. Okay, so here between B and VG, which is what we were looking at in that picture a few slides back, there's a 17% crossover or recombination frequency, which is what was calculated at the bottom of all of that jazz, right? There was a 17% recombination frequency, which means that there are 17 units between B and VG. Well, there was another set of studies done using the trait CN, which is like called cinnabar, and it's kind of like a more orangey eye color. It's not white, it's not red, it's kind of somewhere in the middle. Okay, so remember that this is talking about the body color, this is talking about the wings, and now this is talking about like the eye color, okay? So our eye color here is showing us that we have a 9% recombination with our B gene, which means that they are 9 units apart. And then our VG gene shows a 9.5% recombination with the cinnabar gene. So we know that they are 9.5 units apart. So that's how we know to arrange these genes in this fashion. For instance, you could not have... erasing. You could not have VG here and CN here. This would be like trial and error when you're trying to figure out, did you do it correctly? Because you know from calculations that there is a 9% or 9 unit difference between B and CN. And you know from calculations that there is a 17 unit difference between our B and our VG. So this math, like this here, does not add up, right? So that's how you have to know that you have our CN and our VG in the positions that they are relative to our B trait, right? If this is 17, you know that they have to be farther apart than these two, right? Or than these two, because that's just how math works out, right? 17 is larger than 9. That's how you know that, okay? So I hope that makes a little bit of sense. So genes that are far apart on the same chromosome can have a recombination frequency near 50%. Such genes are physically linked but genetically unlinked and behave as if found on different chromosomes, okay? Because they often can be recombined and crossed over. So they're so far apart that they behave as if they're on different chromosomes because they can't be split away from that one because they are so far apart. So this other scientist, no, I don't know how to say his name, used recombination frequencies to make linkage maps of fruit fly genes. Using methods like chromosomal banding, geneticists can develop cytogenic maps of chromosomes. So cytogenic maps indicate the positions of genes with respect to the chromosomal features. We're going to be focusing on linkage maps in my class personally. Okay, so this is an example of our cytogenic map. Includes the position of genes with respect to chromosomal features. Okay, so we know that we've got these particular traits that we've looked at before, but then he added in some other mutant phenotypes, which you can see here. And based on the frequencies of recombination, the calculations that were done there, we're able to kind of like say that these are the two extremes of our mutant phenotypes. Okay, so alterations of the chromosome number or structure can cause genetic disorders. Makes sense, because if you're missing chromosomes or you have too many chromosomes, you'll either have not enough information or way too much information. It's going to cause some sort of large-scale havoc on the body. So, like I said, large-scale chromosomal alterations in humans and other mammals often lead to spontaneous abortions or miscarriages. It's actually the reason why there are miscarriages. You know, some people like to shame mothers for that, but it has literally nothing to do with them and everything to do with the genetics. If the DNA is not in there and it's not correct, then the body just gets rid of it before anything really comes of it. And that is, you know, a mechanism to make sure that we have strong offspring, or cause a variety of developmental disorders. So plants tolerate such genetic changes better than animals do. Plants can actually have all kinds of crazy mutations. The dogs are fighting in the background again. Sorry about that. Plants can actually live with a whole bunch of weird genetic abnormalities, and they're kind of okay with it, whereas animals, being a little bit more complex in structure and function, don't handle it so well. So we're going to look at some of the examples in animals, specifically humans. So in non-disjunction, you have pairs of homologous chromosomes that don't separate normally during meiosis. So this means that the cells that are produced, the gametes that are produced are not going to have the right number of chromosomes. So then when they are either fertilized or do the fertilizing, there's still not going to be the right number of chromosomes. So that individual is going to have either a surplus or a deficiency of DNA. As a result, one gamete receives two of the same type of chromosome and another receives no copy. So here's what that looks like, non-disjunction. So it's a failure of the chromosomes to separate, which you can see down at the bottom that's kind of a huge deal. Like you should have our N number. You should have that in our haploid cells. Remember, these are haploid because we're in meiosis here. Okay. You should have N chromosomes. N minus 1 means that we're missing one, and N plus 1 means that we have one too many chromosomes. So non-disjunction of homologous chromosomes in meiosis 1 is represented here, and then non-disjunction of sister chromatids in meiosis 2 is represented on this as well. Okay, so you can see that you have N plus 1 in these cases, N minus 1, so none of these have the normal chromosome number, whereas over here at least 2 of them have the correct chromosome number. So aneuploidy results from the fertilization of gametes in which non-disjunction has occurred. The offspring have an abnormal number of a particular chromosome. Okay, so aneuploidy is when we have non-disjunction that has occurred. So like when we have those 3 chromosomes meeting up with, let's say, another 3 chromosomes. Well, that's not, obviously we don't have 3. I was just using this one and this one as an example, right? That's not the correct number of chromosomes to have. So this is going to be an aneuploidy because it is a fertilized non-disjunction gamete here. So a monosomy, trisomy, these are 1. A mono is going to be 1. It's going to be a zygote that has only 1 copy of a particular chromosome, whereas a trisomic zygote is going to have 3 copies. Okay, if it was di, it would be normal, so that's why that's not a thing. We went from 1 to 3 because you either have 1 less or 1 too many. So monosomy and trisomy here. Polyploidy is a condition in which an organism has more than 2 complete sets of chromosomes, like a triploidy or triploidy, which means it has 3 sets of chromosomes, and a tetraploidy, which means it has 4 sets of chromosomes. And a polyploidy is really common in plants, and plants handle it very well, but animals don't so much. Polyploids are more normal in appearance than aneuploidies. So polyploidies, they typically have, more of a normal appearance, whereas there's still some sort of disorder happening within their bodies, whereas an aneuploid will typically look as if there is some sort of genetic disorder occurring. Okay, so we're going to look at the different types of chromosome structure alterations that can be caused by all of this. So breakage of a chromosome can lead to 4 different types of changes in our chromosomal structure, which can also lead to other genetic disorders because our chromosome structure is compromised. Before, we're talking about having too many or too few chromosomes. Now, if you actually alter the structure of those chromosomes, it's equally as terrible, right? So you can have deletions. It's exactly what it sounds like. You're deleting a section of the chromosome. Duplications, you are duplicating a section. You are doubling it. You're making a copy of it. Inversions reverses the orientation of a segment of the chromosome. And translocation is going to move a segment from one chromosome to another. This is what that looks like. So deletion, you're literally going to be deleting the D here. So in our product, there is no D. With an inversion, you are going to be switching the segment. So you will get this as your product. Duplication, you have copied this region. So now you have 2 copies of that. Translocation is when you are essentially switching. So from here and here, we have switched these 2 sections. You're switching sections of a chromosome with another chromosome. Okay, so that's why your results look as they do here. So let's look at some human disorders due to chromosomal alterations. So alterations of chromosome number and structure are associated with some really serious disorders that you've probably already heard of, many of them. Some types of aneuploidy upset the genetic balance less than others, resulting in individuals surviving to birth and beyond, but maybe not their full, potential lives. These surviving individuals have a set of symptoms or a syndrome characteristic of a type of aneuploidy. So Down syndrome is trisomy 21. So trisomy means that there are 3 chromosomes in the 21 position where there should only be 2. So Down syndrome is an aneuploid condition that results from 3 copies of chromosome 21. It affects about 1 out of every 700 children born in the United States. And the frequency of Down syndrome increases with the age of the mother, a correlation that has not been explained. So it just happens that the older the mother is, I forgot to open this back up, I'm sorry. The older that the mother is, the higher the probability that the offspring will be born with Down syndrome. Some experts believe that it's just because when a female is born, she has all the eggs in her body she's going to have. So the age of the eggs is equal to the age of the female. So the older the female, the older the eggs. And over your lifetime, you're getting little tiny micro mutations all the time. So that's thought to maybe be a link, but not been proven yet. So here's an example of a trisomy 21, where this is a karyotype that you're looking at. This 21st position has 3 chromosomes there where it should only have 2. Next, we're going to talk about aneuploidy of sex chromosomes. So non-disjunction of sex chromosomes produces a variety of aneuploid conditions. Klinefelter syndrome is a result of an extra chromosome in a male producing the XXY individuals. So you have 1 too many. This is a trisomy again. Females with trisomy XXXX have no unusual physical features except being slightly taller than average. Again, they're just kind of getting extra genes here. And of course, bar bodies would still be at play. Monosomy X, where you only have 1, you have like X and then nothing else is written as a 0, is called Turner syndrome and produces females who are sterile. They also have other physical abnormalities typically, that they just don't look as feminine typically. And it's the only known viable monosomy in humans, which means that they will survive, which is actually, pretty interesting that it's the only one in humans, a monosomy that you'll actually survive. So then disorders that are caused by structurally altered chromosomes, like we were talking about the deletions, inversions, and all of that. So there's a syndrome called cry of the cat, results in a specific deletion in chromosome number 5. A child born with this syndrome is mentally retarded and has a cat-like cry. And individuals die in infancy or very early childhood. So obviously this is it ends up being this like lethal deletion early on in life. And then there's certain cancers, multiple different types of cancers actually, that are caused by translocations of chromosomes. This is an example of a translocation of a chromosome between chromosome 9 and chromosome 21, where they're actually swapping bits of information here to have this result. This is actually called the Philadelphia chromosome because this particular translocation causes cancer, and the first type of cancer, this was first seen in Philadelphia. This particular cause of cancer, this type of cancer that was caused because of this reciprocal translocation, was first seen in Philadelphia. So chromosome 22 is sometimes called the Philadelphia chromosome because of that. Okay, yay, we made it to the end. So my next thing I'm going to talk about is going to be the chi-square. And then, of course, I'm also going to go through and create a video to show you how to do the mono hybrid Punnett squares, the dihybrid Punnett squares. I'm going to show you co-dominance, and I'm going to show you incomplete dominance. I'll also show you sex-linked traits, and I think that about covers it. So thanks for sticking with me, and I'll see you in the next one.